Okay, good morning. Last class we discussed the cell organelles, mainly different types of the cell organelles. Yesterday we discussed the cell wall and next one some of the cell organelles. Especially some of the endoplasmic reticulum, mainly they are the endoplasmic two types, smooth ER and rough ER. Okay, smooth ER and rough ER. And next cell organelle is the nucleus. Nucleus is the large roundish the organelle. It is the bounded by the double membrane, which is the numerous pores inside. And the nucleus also they are contained the chromosomes and dark region called nucleolus, which makes the ribosome. Actually, nucleus is the center part of the cell organelles. And next one is the integrity of the cell organelles. Next one is the disease relevances. Okay, this is mainly the nucleus. At the same time, nucleus also they are containing the four layers. One is the nuclear envelope, another one is the nuclear pores, nucleolus, and third one is the nucleosap or nucellus. The fourth one is the uh, chromatin. Now, nuclear envelope. First one is the nuclear envelope. Nuclear envelope is especially these are the made of the double membrane model. These are the two membrane models, the outer membrane and inner membrane. Outer membrane especially resembles to the endoplasmic reticulum. Inner membrane is resembles to the ribosomes. Inner membrane, outer membrane and inner membrane. Next one, next one is the nuclear envelope. Also, they are containing the nuclear pores. What are the importance of the nuclear pores? Nuclear pores also is the gateway keepers of proteins. Especially, these are the protecting main nuclear envelope. Also, is that the protection mainly is that the protection purpose. Next one is the they can transport of the some of the proteins. Next one, some of the carbohydrates, proteins, and ribosomes also. Next one is the nucleoplasm or nuclear sap. Mainly these are the liquid portion. These are also they are containing the many proteins and the carbohydrates, ribosomes. Mainly these are the ribosomes also they can use for the protein synthesis purpose. Next one. Nuclear. Next one is the third one is the nucellus. Nucellus also is the liquid portion. Mainly is main. These are also they're containing the some of the proteins, ribosomes, all liquid portion. These are the they can protection and at the same time these are the maintain the integrity. Nuclear plasm also they can maintain the integrity. Next one is the last one is the chromatin. Chromatin also they are containing the DNA plus and some of the proteins, especially DNA also double helix and double stranded DNA. Chromatin. Next one is some of the proteins. But what type of the proteins also they can see into the nucleus. Nucleus mainly keystone proteins. Keystone proteins also are the four types of the keystone proteins. H1, H1A, H2A, H2B, different four types of the keystone proteins. What are the main importance of the nucleus? It is the controls the all metabolic functions of the cell integrity. The control of the controls all the metabolic functions of the cell. It helps the cell division. It helps the cell division. It helps the protein synthesis. It helps the in cell cycle. It is responsible for the transfer of hereditary and the characters from the parents to next generation. It is a brain of the cell, especially nucleus also is the brain of the cell. The okay, plasma membrane already we discussed yesterday. Cytoplasm also mainly is the fluid content present inside of the plasma membrane. It contains the specialized cell organelles function. It acts as the store of the important chemicals. It is the physical base of the all metabolic activities. It keeps the cell fully expanded and the provides the turbidity. Okay, nucleoplasm, sorry, cytoplasm, endoplasmic reticulum. Endoplasmic reticulum is a large network of membrane-bounded tubes and sheets. It looks like long tubules or bags. It is the structure is similar to plasma membrane. It encloses the fluid filled the lumen. There are two types of the endoplasmic reticulum: smooth endoplasmic reticulum. They do not have the ribosomes in the smooth endoplasmic reticulum. Especially these are the synthesis of the liquids. Rough endoplasmic reticulum, mainly ribosomes are attached to the its surface. ER, what are the functions of the ER? It is associated with the synthesis, storage, and the transport of the metabolic products. It gives the internal support to colloidal matrix. Rough endoplasmic reticulum is concerned with the protein synthesis. It helps in the formation of the new cell membrane, endoplasmic reticulum. Next one is the ribosomes. Endoplasmic reticulum also main function in detoxification process. Detox means the enzyme that the inside the lipid components or lipoproteins are the located in the membrane of the smooth ER. It contains the enzyme catalase series reaction the detoxify the both lipid soluble drugs and various harmful compounds produced by the metabolism. The most extensively studied of the detoxification reaction and catalyzed by the cytochrome P450 family of the enzymes. These are very important. P50 family of enzymes also they're containing the they can use for the detoxification of the drugs. And next one is the next release and reuptake and the storage of the calcium ions. Next one, ER is the most eukaryotic cells and sequester of the calcium from the cytosol. The reserve the calcium to the cytosol from the endoplasmic reticulum. Its subsequent reuptake medicines many rapid responses to extracellular signals. The storage of the calcium in the ER lumen is the facilitate the high concentration of the calcium and binding the proteins there. In some types of the specify the reaction endoplasmic reticulum specific to calcium storage muscle cells. For example, in the abundant specialized the smooth ER called the sarcoplasmic reticulum, which is the request the calcium from the cytosol by the means calcium plus ATP. The pumps indicate the calcium ions. Exchange and translocation. Another important character is the exchange and translocation. Diffusion as well as the active transport may regularly take place through the membrane system. The presence of the carriers and the permeables has been the postulated to help the endoplasmic reticulum membrane system to perform its the function. Endoplasmic reticulum may also act as the kind circulatory system for intracellular circulation of various substances. It may help the translocation substance both cell interior and the exterior. Protein synthesis, mainly rough ER also they can use for the protein synthesis. Some protein molecules also synthesized on the attached ribosomes are the discharge and penetrate into the cavity of endoplasmic reticulum. These are the either stored or the exported to the outside. Next one is the ribosomes. What are the importance of the ribosomes? Ribosomes are the densely spherical granular particles which move freely in the matrix. They are the present in cytoplasm as well as the inside of the nucleus. A ribosomes is made of the numerous proteins and the RNA. Mainly, these are the made of the numerous proteins and the RNA ribosomes. Two types of the ribosomes actually, 80S ribosomes and 70S ribosomes. 70S ribosomes, especially they are the present in the in prokaryotes. What are the subunits of the ribosomes? In 50 plus 30 subunits, S means Sedberg units, and 80S types of the ribosome, especially they can is are also seen to the in eukaryotes. What are the functions of the ribosomes? Mainly, they play the important role in protein synthesis. It helps the storage, modification, and packing of the products in vasculature. It is involved in the synthesis of the lysosomes, especially involved in the synthesis of lysosomes. Golgi bodies, apparatus, structure. It consists of the smooth, especially Golgi bodies in the apparatus. What are the structure? It consists of the smooth, flatted cisternae. They are usually together in parallel rows. Their their membranes have the connection with the membrane of the endoplasmic reticulum. The Golgi complexes looks like a stalk of the snacks made of the membrane with the smaller vascules on the other side of the main structure. What are the functions of the Golgi bodies? Golgi apparatus mainly first one discovered by the Camillo Golgi. It is the is also called as the dictyosomes. Dictyomin network appearance cells. Okay, mainly the Golgi. Each Golgi stacks as to the two distinct phases. One is the entry phase, sys phase. Another one is the trans phase, exit phase. Mainly, uh, fen- fenestrated lamella is the core or more opening spores also they can present into the in Golgi operators. They can ask into the in competitive examination point of view, especially fenestrated lamellae core or more openings or pores also they can see into the in Golgi operators. Mainly, chemically the mainly contain of the lipoproteins and two main enzymes, thymine pyrophosphatase and glycolol transferase. These are the two protein the enzymes also they can see into the Golgi apparatus. The very important function Golgi complete transport between the cells and within the cells, intracellular and intercellular, and refer the membrane trafficking, especially Camillo Golgi. Okay, what are the functions of the Golgi operators? It is involved in the synthesis and the protein liquids carbohydrates, synthesis of the proteins, liquids, carbohydrates. It is involved in the process of the lysosomes and peroxisomes. It complex sugars from the simple sugars. It packages and dispackages the material synthesis near the targets inside of the outside of the cells. It helps the dispackage and the material synthesis near targets inside and the outside of the cells. It helps in the storage, modification, and packing of the products. Okay. Next one is the lysosomes. There are the small spherical bodies bounded by the single membrane. Especially the single membrane models are the lysosomes, peroxisomes. Okay, these are the single membrane models. They contain the lysosomes, also digestive enzymes, which break the all organic materials. They act the digestive for the cell, so they are the also called digestive bags. The cause of the digestion for the one outside of the organelles and the even can digest entire damaged cells containing them. They are also called as the suicidal bags. What are the functions of the lysosomes? They help the intercellular and the intracellular digestion of the food particles. Okay, they cause the digestion of the one but and the damage of the cell organelles. They destroy the foreign substances. They help the digestion of the bones and the digestive. Next one, vacuoles. There are the solid sacs for the solid or and liquid contents. That the small size animal cells and the large cell in plant cells, especially vacuoles also only they can see into the in most of the plant cells. What are the functions of the vacuoles? They help in the storage of the food, water, and substances. Contractile vacuoles help in the eliminated excess water from the soil. Next one is the mitochondria. Mitochondria is the structure. Mainly these are the developed the benda mitochondria. They are the rod-shaped structure bounded by the double membrane, and outer membrane is very porous while the inner deeply folded. Especially outer membrane is the uh, very porous, especially pores are present in the outer membrane. Inner membrane also with a very uh, mm, inner deeply in the folded. This is also called as they can form the cristae. While inner deeply folded, the folds create the large surface area for ATP generating chemical reaction. They are called strange organelles because of they have their own DNA and ribosomes. Therefore, they are able to make some of their proteins. It is also called as the powerhouse of the cells because they release energy required for the various activities in the cell. Mitochondria. Actually, mitochondria the first reported in 1818 is the Benda. Okay, these are organelles are usually spherical, ovoid in the shape. They may be the filamentous, and next one granular, club-shaped, vascular, rod-shaped. These are the distributed throughout the cell volume, and they may have more or less definitive orientation. Cylindrical cells, they are the generally oriented in the basal apical direction, parallel to the main axis. In leukocytes and mitochondria are arranged the radially with respect to the centrioles. It is believed the orientation depend on the submicroscopic organization of the cytoplasmic matrix. These are the actually the length is the 0.2 from metric to cells 40 mm, especially the amphibian oocytes, the bigs in the 40 mm. Okay, the size of the influence the both developmental stage of the cell and other physical environment within the cell, such as the pH, osmotic pressure, and cell activities. When the living cells were observed under the microscope, the mitochondria appear to be continuously and moving, and the changing of their shapes and the cytoplasm depending on the inner constraints and the external environment. In general, their number of ranges from the to maximum 3 lakhs in oocytes, and that the number of um, diminishes of concerned tissues due to the increase the aerobic glycolysis. Ultrastructure, mainly ultrastructure is the double membrane model, two membrane highly specialized membranes. The two separate mitochondrial components, the internal matrix space and the much narrow intermembrane space is also called as the pre-mitochondrial space. Okay, is the outer membrane is the outer membrane thick is the 6 nanometer thick. The contain the channel formatting proteins called is the porins. These are the responsible for the transport of molecules of 5,000 daltons or less, including the small proteins. Such molecules can enter into the intermembrane space, but most of them cannot pass the impermeable inner membrane. Thus, the intermembrane space is the chemically equivalent to the cytosol with the respect to the small molecules, while the matrix space contains the highly selected small molecules. Inner membrane. The inner membrane also 6 nanometer thick and the folded into the numerous cristae, which is generally increase the total surface of the area. It contains the high proportion of double phospholipid cardolian, which contain the four fatty acids. This makes the membrane impermeability to ions. It also contains the variety of transport proteins that make selectively permeable those small molecules that is the metabolized or required by the many mitochondrial enzymes in the matrix space. The membrane proteins perform the three functions. Those that the carry out the oxidation reactions of the respiratory chain. And next one is the enzyme complex called is the ATP synthase that makes ATP in the matrix. The specific transport proteins that is the regulate the passing metabolites into the outer membrane. And inner membrane space, which is the about the 6 to 8 nanometer thick contain several enzymes that is used the ATP passing of the matrix and the phospholipid the outer membrane. Matrix space. The matrix space contain the high concentrated mixture of the unrates of the enzyme, including those require the oxidation of pyruvate and fatty acids. And for the citric acid cycle, the matrix also contains the several identical copies of the mitochondrial DNA genome and the special mitochondrial ribosomes and tRNA and various enzymes required for the expression of the mitochondrial genes. The inner membrane is usually highly highly convoluted forming the series of the enfolding known as the cristae. The inner matrix space. This then convolution the greatly increase the area inner membrane such as the attains the size of the 1 by third of the total area is the membrane. The membrane cristae variable with the type of the cells. In foldings, the tubular plants are known as the tubulae or microvilli. The frequently plants intermediate types of the folding are also seen. Next one, elementary particles, mainly in mitochondria attached to the some of the elementary particles. The inner membrane substance, the oxysomes. These are the particles 85 angstroms diameter, and they regulate the spatial intervals of the 10 nanometer on the inner membrane. Then we also there are also F1 particles. They are also called as the F1 particles or oxysomes. When the mitochondrial cristae are the dispersed, the sonic vibration, the submitochondrial particles are obtained from the oxysomes are seen into the attached to the outer surface. These submitochondrial particles are the responsible for the respiratory chain and the phosphorylation. Okay, well, especially formation of the ATPs. They are the main. They can the oxysomes have been the identified the molecules ATP is enzyme. The responsible for the catalyzing the terminal step of the ATP. The mitochondria may be altered due to the action of the various agents, but most of these changes in the reversibly. Only of these the changes reach in the certain critical point, they become the irreversible. Okay, sometimes the mitochondria is in the place of the autolysis, forming some kind of the lysosomes called cytolysozymes. Mitochondria may also undergo the degeneration due to the fusion and forming a large bodies is called as the chondriospheres. Chemical composition of the mitochondria. Mitochondria can also uh, isolate the cell fraction brought about the different centrifugation. Mainly the chemically mitochondria mainly consist of the protein 65 to 70%, and the lipids also 25 to 30%. And next one is the both are the together they can form to the 90 to 98%. Other remaining particles are the DNA, RNA. They're containing the many enzymes. Okay, mitochondria mainly are the it has found that the liver mitochondria is 60% of 67% of the total protein matrix, 21 located in the inner membrane, while the 6% each of the located their outer membrane and membrane space. These are the mitochondria have the own protein synthesizing machinery, including the ribosomes. What are the functions of the mitochondria? And there are also powerhouse of the cells. Mainly powerhouse of cells mean during the breakdown of the substrates into the ADP to produce the ATP. This mitochondria were viewed as the powerhouse of the cells. Okay, next one is the role of glucogenesis. Gluconeogenesis, conversion of the non-carbohydrate to glucose form. Uh, glucose form glucose from pyruvic acid. Non-carbohydrate, especially pyruvic acid, is the mainly glucose from mainly non-carbohydrates to glucose from pyruvic acid. The pyruvic acid one formed the converted to the oxaloacetic acid. Okay, role of amino acids in the lipid metabolism. Next one is the biogenesis. Biogenesis is the Biogenesis also is the undergo the process where the mitochondria produced the still incomplete. Levenger and the recognize the possible to reduce their function. Formation from the other membrane structural in the cell, pinching in the growth and divisions of the pre-existing of the mitochondria. Deonovosynthesis. The mainly deonovosynthesis also is very important. In the early part of the century, the experimental evidences were known for the kind of origin of mitochondria. Deonovosynthesis also they can, for example, sea urchin eggs were the freed from the mitochondria before development. At the development, larvae have the sufficient number of the cells. Next one, evolutionary origin of the mitochondria, monophyletic origin from the free living proteobacterium, especially they are formed to the proteobacterium. Simultaneous origin of the eukaryotic cells and the mitochondria. Okay, these are the mitochondria in functions and structures. When they are the second largest cell organelles in the unity of the genetic structure. The double layer outer membrane inner folds the cristae. Energy producing chemical reaction take place, controls levels of water and the other materials in cells, recycles and decompose the proteins, fats, and carbohydrates from the urea. Next another cell organelle is the plastids and the chloroplast. Chloroplast. The first one is the Schimper. Chloroplast and plastids. The first one used in the Schimper. Okay, is are also bimembrane organelle, mainly found in the plant cells, some of the algal cells. Mainly the responsible for storage, the wide variety of products, the process of the photosynthesis and synthesis of the some key molecules necessary for the cell architecture and function. Depending on the presence of or absence of the pigment, Schimper is classified the plastids in the three main types. Okay, these are the Schimper. They can divide the plastid into three types. One is the leucoplast. Leucoplast mean white color. Chromoplast. Chromo mean color. And chloroplast. The chloro is especially green. Chloroplast is a very important, playing the crucial role in the green cells. All types of the plastids are the developmentally related to the all types of the plastids are developmentally related to the precursor structure is also called the protoplastids. The internal milli of the protoplast appears in the uniformly dense and finally granular and is also called as stroma with a few lamella and vesicles. In this unit, we is the focus our attention of the green colored plastids. Chloroplast is also chloroplast. Chloroplast is the very shape and size and the number of different species and is also different cells within the species. The shape of the chloroplast is highly variable. In all higher plants, the shape is the generally spheroid, discoid, or ovoid shape. Many examples of the variation in algae, some of the algae, some these are the cup-like shape, in the spiral band-like reticulate and ribbon-like structure and disc-like structure. Also, they can see into the most of the algal members. Most of the higher plants cells that is the biconvex and the measure of the 4 to 8 millimeter in diameter and 2 millimeter in thickness. The number is also variable in the various species. Many species of the algae, again, the chloroplast may be one or two. Many for cell, there is the most of the higher plant cell containing the different members of the chloroplast. Okay. Next one is the ultrastructure of the chloroplast is the outer membrane, envelope membrane. Next one, inner envelope membrane and grana and stroma. The outer envelope membrane also it contains the non-specific pore proteins. There is the freely permits the water and variety of ions and metabolites of size up to the approximately 10 kilodaltons to pass into the aqueous space between the outer and the inner envelope membranes called as the intermembrane spaces. The outer membrane is the smooth and never becomes conflict. with the inner membrane in any point. The electron microscope studies have relieved the contact sites where the proteins connecting the membranes mediate the transfer of the nuclear encoded the chloroplast proteins from the cytosol, the stroma. All the enzymes involved in the galactolipid metabolism is localized the outer membranes. The inner envelope membrane, outer envelope is the chloroplast is called so the inner envelope membrane. It is freely permeable the small uncharged molecules including the O2, ammonia, and many low molecules mass carboxylic acids. Mainly the transport of the many of them require the specific transport proteins. It also contains the enzymes involved in the assembly of the thylakoid membrane lipids. This means the grana and stroma network. As stated earlier, the proplast themselves contain the uniformly dense in the finally granular substance inside termed as the stroma. And during the chloroplast differentiation from the proplastids in the presence of the light in the highly complex membrane system develops in the stroma is also called the thylakoid membrane within the chloroplast. The thylakoids form continuous network enclosing the single on stroming chambers. The thylakoid network consists of the two distinct types membrane domains. The stalked grana, these are the singular grana. The thylakoids are the unstalked stromathylakoids. There are later two interconnect of the grana stalks through the chloroplastoma. A given membrane can be stacked. Okay, grana and stroma networks. What are the functions of the chloroplast mainly? The functions of the food, mainly food through the photosynthesis. They provides the colors and the fruits and the flowers attached to the for the pollination. It stores the starch and the oil and protein granules. The chloroplast. Ribosomes already is are not membrane bounded are the ribosomes. Mainly these are the 80s and 70s ribosomes. What are the chemical composition of the ribosomes? Ribosomes are the made of the ribosomal RNA, different types of proteins. The molecular weight range from 12,000 to 25,000 daltons. 80s ribosomes possess only molecules 25 to 25 RNA, which hydrogen bonded is the 5' S RNA, one molecule of the 18S rRNA, and the one molecule of the 5S rRNA. 70S ribosomes have the 23, 16, 5S RNA functions. Mainly, these are the uh, magnesium ions that play major role in the structural organization of the ribosomes. Ribosomes also the main functions is the protein synthesis, especially ribosomes are cell organelles where the translation of the genetic code carried by mRNA occurs, leading to the formation of polypeptide. Next one is the plasmodesmata. The tissue plant are the organized differently from the animal cells, mainly because of their cell walls. The cell walls rich in the cellulose helmet need for the anchoraging junctions hold the cell's place. Plasmodesmata is mainly are the seen to the animal cells. Plant cells have only one class of the intracellular junctions, plasmodesmata, like the gap junction, mainly gap junction of the animal cells directly connect the cytoplasm and the adjacent walls. In most plasmodesmata, there is the narrower and cylindrical structure in each, and desmotable running throughout the center of the channel. It is continuous with the elements of the smooth endoplasmic reticulum in each of the connected cells. Between the outside of the desmotubule and the inner face of the cylindrical channel formed by the plasma membrane. The inner face of the cylindrical channel formed the plasma membrane is the annulus cytosol, through the which small molecules can pass through the cell to cell. Plasmodesmata are the normally created a new cell walls formed by the after cytokinesis of the cell division. They form around the elements the smooth air that the becomes trapped across the developing of the cells. Plasma membrane is the surround the cylindrical strand in the tightly fluid endoplasmic reticulum. The desmotubules are oppressed are the cell organelles. Okay, next topic chromosomes. Chromosomes and structure and functions of the chromosome. Mainly, E. Strasberg in 1875 first discovered the thread-like structure appeared during the cell division. These thread-like structures were the called chromosome due to the affinity of the bases dyes. The term chromosome is derived from the two Greek words, chromos in color and somum in body. This term was first used by the Walder in 1888. Chromosomes contributed to the division of cells, and they are the prime importance as they carry the genes which are the heredity material. Chromosome number of chromosomes has given the species generally constant. All the members of the species ordinarily have definitive and the generally constant somatic and gametic chromosomes number. Some chromosomes number is the number of chromosomes found in the somatic cells of species and the represented by the 2n. Generally, somatic cells have the contain two copies of the each chromosome except the sex chromosomes. Both both the copies are the ordinarily an identical internal morphology gene contain and the gene order and the it's known as the homologous chromosome, same chromosome, homologous. Heterochromosome, different types of the genetic chromosome number is exactly half of the somatic chromosome number and is represented by the n, 2x and 2n are n. It denotes the number of the chromosomes found in the gametes of the species n condition. Chromosome size. The size of the chromosome shows the remarkable variation depending upon the stage of the cell division. Longest and thinnest during the interphase and the ends not visible under the microscope. Longest and thinnest during the interphase and hence the visible visible under the microscope. Smallest and thickest during the mitotic metaphase. A long and especially long side of the chromosome they can see into the in interface, and short the smallest thickness during the seeing the metaphase. Chromosome size is not proportional to the number of genes present in the chromosomes. Chromosome morphology. The outer covering of the sheath of the chromosomes is known as the pellicle, which encloses the matrix. Within the matrix lies the chromatin. Already we discussed the chromatin mean DNA plus some of the histone proteins is also called as the chromatin. Okay. Fleming reintroduced the term chromatin in 1879. The chromosome morphology changes during cell division and mitotic metaphase is the most suitable stage for the studies in the chromosome morphology. In mitotic metaphase chromosomes, the following structural features can under the light microscope: chromatid, centromere, telomere, secondary construction, chromomere, chromonema matrix, chromatid. Each metaphase chromosome appears to be longitudinally divided into the identical parts of the each called the chromatid.
Hello, sir. Good morning. Hello,
Chromatid. Each metaphase chromosome appears to be the longitudinally chromatid. Each metaphase chromosome appears to be the longitudinally divided into two identical parts, each which is also called as chromatid. Chromatids of a chromosome appear to be joined together at the point known as the centromere. See, in between the chromatids, especially in the central part, is also called as the centromere. Two chromatids make up the chromosomes are the referred to as the sister chromatids. The chromatids are the homologous chromosomes known as the chromatids of homologous chromosomes are known as the non-sister chromatids. See the appears the outer structure sister chromatids, the non-homologous chromosome also non-sister chromatids. Okay, the centromere, the region where the two sister chromatids appear to be joined during the Mitotic metaphase is known as the centromere. It generally appears as the construction and the ends called the primary construction, helps in the movement of the chromosomes to opposite poles during the anaphase of the cell division. The centromere consists of two disc-shaped bodies called kinetochores. Normally, chromosomes are the monocentric, having one centromere each. Next one is the depending of the position of the centromere, chromosomes are the grouped as metacentric. Centromere is located in the exactly to the center of the chromosome. It is also called as the metacentric. Its shape is the V shape at anaphase. Submetacentric. The centromere as the located on one side of the centromere point, such that one arm is longer than the other. These chromosomes become J or L shaped anaphase. Acrocentric. Centromere is located closely to one end of the chromosomes, and thus the given the very short arm and very long arm. These chromosomes acquire the J shape or the rod shape of during the anaphase. Telomeres, centromere is located at one end of the chromosome, so that the chromosome has only one arm. These chromosomes are the I-shaped or rod shaped. Is a telocentric. Telomere. Two ends of the chromosomes are known as the telomeres. They are highly stable and do not fuse to the unity of the telomeres chromosome due to the polarity effect. Any broken end of the chromosome is unstable and can join with the piece and another chromosomes. But the telomeres import the stability to the chromosomes, which retain its the identity and individuality through the cycle and the generations. Secondary construction. The constructed of are narrow regions other than the centromeres is also called as the secondary construction. The, the chromosomes having the secondary construction are known as the satellite chromosomes or SAD chromosomes. Chromosomes may possess the secondary construction in one or both arms of it. Chromosomal ends distal and to the secondary construction is known as satellite. Production of the nucleus is associated with the secondary construction, and therefore it is also called as the nucleus organizer region. Satellite chromosomes are often referred to the nucleus organizer chromosomes, chromomere. In some species like the maize, ray, etc., chromosomes, the packing stage meiosis show the shell. Bed-like structure is also called as the chromomeres, bed-like structures. The distribution of the chromomeres, the chromosomes is highly characteristic and the constant. The pattern of the distribution being different for the different chromosomes. There are clearly visible as dark staining bonds in the giant salivary and gland chromosomes. Chromosomes are the region of tightly and the folded DNA. Chromonema. A chromosome consists of the two chromatids, and each chromatid consists of the thread-like coil structure called the chromonema. The floral chromonemata. The term chromonema was coined by the Wedgeski in 1912. The chromonemata from form of the gene bearing the portion of the chromosome matrix. The mass of the achromatic material which is the surrounds the chromonemata is also called as the matrix. The matrix is enclosed the sheath which is the known as the pellicle. Both the matrix and the pellicle are non-genetic materials and appear only at the metaphase when the nucleus disappears. Composition of the chromosomes. Mainly, the material of which chromosomes are composed called chromosomes. And Fleming introduced the term is the chromatin in 1879. Chromatin was the classified the two group in the cytoplasm and the affinity of the basic dye like estocaramine and or Pelgin basic dyes in the basic fusion in the reagent at the prophase. The darkly stained regions were the called the heterochromatin, while the tightly stained regions are also called as the euchromatin. Darkly stained Okay, darkly stained regions also called as the heterochromatin, and lightly stained regions also called as euchromatin. The differentiating capacity of the different parts of the chromosomes is known as the heterokaryopsis. Heterochromatin further classified into two groups: constitutive and the facultative composition of the chromosomes. Mainly is the karyotype and ideogram. The characteristic features of the by which a set of chromosome or species is identified. Generally, karyotype represented by the arranging of the chromosomes in descending order, size, keeping the centimeters in the same line. The karyotype of the species can be represented diagrammatically, showing the all the morphological features of the chromosomes. Such a diagram is known as the ideogram or ideotype. Next one, special types of the chromosome. Some tissues certain the organism contain the chromosomes which are the significantly from the normal chromosomes in the terms either morphology or function. Such chromosomes are referred to the special chromosomes. The following under the categories: giant chromosomes or polytene chromosomes. This is where the first discovered the E.G. Balbani in 1882 in diphtheria salivary glands, and hence the commonly called salivary gland chromosomes. Okay, giant chromosomes. These chromosomes replicate the reptile, but the daughter of the chromatis do not the separate from one another, and the cell also does not the divide. This is the phenomena. The phenomenon is known as the endomitosis or reduplication. It results in the formation of many standard giant chromosomes known as the polytene chromosomes, and the condition is known as the polyteny. The size of the is 200 times or more than the normal somatic chromosomes, autosomes, and the very thick, and the known is the giant chromosomes. The special type of the chromosomes, special types of the chromosome, Lamb-Rush chromosomes. These were the first observed in the W. Balban in 1882. We discovered the detailed oocytes of the shark and croquettes in 1892. They occur at the diploidine stage of the mitotic pherophase oocytes of the animal species. Since they are found in the mitotic prophase, they are present in the bivalents in which the maternal and the paternal chromosomes are held together by the chiasmata, those sites where the crossing over as previously occurred. Each bivalent has the chromatids 2 in each homologue. The axis of the each homologue consists of a row granules are the chromosomes, each of which is have the two loop like lateral extension are formed in each chromatid. Thus, each loop represents the one chromatid in the chromosomes and is the composed one DNA double helix. One end of the each loop thinner than the which is known as the thickened. There is the extensive RNA synthesis ends of the loop, while there is the little or no RNA synthesis attacks. Special type of chromosomes, the accessory chromosomes. In many species, the chromosomes found in the addition of the normal somatic chromosomes. These extra chromosomes are called accessory chromosomes or chromosomes, supernumerary chromosomes. These chromosomes are the broadly similar to the normal somatic chromosomes in their morphology, but have some peculiar functional aspects. For instance, the presence of the several such chromosomes are the often lead to the reduction of the vigor and the fertile in males. These chromosomes are generally smaller in size than the normal spermatic complement. Origin of the this chromosome most species is unknown. Isochromosomes which is to the identical each in their the gene content and the morphology. Allosomes and sex chromosomes. Different morphology in the number male and the female is also called as allosomes. They're responsible for the determination of the sex. XY chromosomes in the human beings and the Drosophila allochromosomes, sex chromosomes. Chromosomes have which have no relation with the determine the sex and the contain genes which determine the somatic characters of the individuals are called autosomes and are the represented by the letter A, special types of the chromosomes. Some of the models of the DNA folding, mainly they can discuss the nucleosome models, solenoid models. The solenoid model is very important. Nucleosome model, the nucleosome model also they can see into the electron microscope studies the chromatin from the interface of the nuclei. Lies in the water have shown the chromatic fibers with arrays of the spherical particles of the 10 nanometer as they repeating units which occur the regular intervals of the given species. It is also called as the nucleosomes. These nucleosomes are the fundamental units of the chromatin in all the eukaryotes. When the nuclei are the lies the low salt solution, the chromatin decondenses and the nucleosomes can be observed the resembling bands and the string, electrophoretic and the gel filtration. The techniques have the relieved the existence of the five histone proteins H1, H2A, H2B, H3, H4 represents the present the chromatin eukaryotes involved in the organization of the nucleosomes. With the use of the chemical cross-linking of the histone proteins, it is found in the H3 and H4 are present in the trimer. And next one, while the H2A and H2B is the associated to the another trimers, and together this is the true trimers from the octometer is also called as the core particle. Known length of the DNA wraps around this the core particle. H1 H1 histone protein as seen outside of the core particles. The DNA that the present between each adjacent core particles by from the 18 to 114 base pairs and this is also called the linker DNA. The kind of the nucleosomes model the wrap DNA around the core Okay, solenoid, different types of the models. Solenoid model, and next one is the nucleosome model. Solenoid model also is the later shows 11 nanometers fiber nucleosome get the coil upon the itself to form the 30 nanometer by the helix with the 5 or 6 nucleosomes per helix. In this helix, successive turns become close to one another. There is a center distance about the 10 nanometer. This 30 nanometer structure was called the solenoid. The term is fixed to the called the central axis, solenoid and loops. Okay. Next one, E chromat, endochromat already we discussed. Okay, these are the chromosomes. Thank you.